Welcome to MRU E-Learning, powered by Malla Reddy University. This is Dr. Haripriya from Department of Biochemistry, School of Allied and Healthcare Sciences. Today we will have a discussion about the introduction of biochemistry, which explores its journey from molecules to the life. Biochemistry is a branch which deals with the metabolic reactions happening in the living system. So exactly, we are discussing about the role, function, and structure of biomolecules, where we are going to discuss about its origin, formation, function, and what are the deficiencies which are associated with these biomolecules, and what are the symptoms. Now, without functioning of chemical reactions, there is no life in any form. In humans, at the most basic level, the proper functioning of all the metabolic reactions are responsible for good health. If there is any disruption in any of the metabolic reactions, obviously it leads to one of the metabolic or genetic disorders. So, the study of biochemistry is very important for deepening of our understanding of how an organism functions. So, the term biochemistry was launched by the scientist Carl Alexander Nuremberg in the year of 1903. This is derived from a Greek word called bios, which means life. This subject is very important, which helps us to understand about the structure and functions of biomolecules, which includes your carbohydrates, proteins, amino acids, lipids, and nucleic acids, and their metabolic pathways, and the disorders which are associated with this metabolisms and the regulation of their biological process. Biochemistry is useful in various applications like medicine, agriculture, nutrition, biotechnology, forensic science, and pharmacology. As we are rapidly going in a newer generation, there are different branches of biochemistry. First one is structural biochemistry, wherein we are going to discuss about the structure and functions of the biomolecules, metabolic biochemistry, all the metabolic reactions associated with the biomolecules, molecular biology, where we are going to study about the biochemistry in terms of molecular level, enzymology, where all the metabolic reactions are going to take place with the help of enzymes, endocrinology, which is a study related to the endocrine glands, bioinformatics, clinical biochemistry, immunology, which is also a part of microbiology, neurochemistry, plant biochemistry, and environmental biochemistry. So these are all the new branches which have been developed from the parent biochemistry. So just like cells are the building blocks of tissues, biomolecules are the building blocks of cells. So now we are going to discuss in depth about the structural biochemistry, where we are going to discuss about the biomolecules. So these are the essential organic molecules which are involved in the maintenance and metabolic process of living organisms required for sustenance of life. So basically we have four types of biomolecules: carbohydrates, proteins, nucleic acids, and lipids. So each of these types of molecules are the polymers that are associated from single units called monomers. So each type of macromolecule is an assemblage of different type of monomer. Like carbohydrates are formed by the association of monosaccharides, lipids are the combination of fatty acids and alcohols, proteins are formed by amino acids, and nucleic acids with the structure of nucleotides and nucleosides. Now let us have a brief discussion about the classification of carbohydrates. Before that, carbohydrates are defined as the polyhydroxyaldehydes or ketones which produce them on hydrolysis. So these carbohydrates are classified under three categories: monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides will have only one sugar molecule in its structure, which are simple, sweet in taste, and easily soluble in water. And they are classified into two types based on their functional groups, aldose and ketones, and based on the number of carbon atoms. So at least we require 3 carbon atoms in their structure. Coming to oligosaccharides, you will have 2 to 10 sugar molecules in its structure, and these monosaccharide units are interconnected with the help of glycosidic bond. Coming to oligosaccharides, if we have only 2 monosaccharide units, we call it as disaccharides. If we have 3 monosaccharide units, we call it as trisaccharides. And if we have 4 monosaccharide units, we call it as tetrasaccharides. Now coming to disaccharides, again it is classified into 2 types, reducing disaccharides and non-reducing disaccharides. This is based on the free functional group. If the functional groups are free, we call it as reducing disaccharides, wherein your examples are maltose and lactose, and non-reducing disaccharide, wherein the functional groups are involved in the glycosidic linkage. There, your example is sucrose. Now coming to polysaccharides, where it has more than 10 monosaccharide units in its structure. And it is again classified into 2 types, homopolysaccharides and heteropolysaccharides. If all the 10 monosaccharide units are of same type, we call it as homopolysaccharide, and the examples include starch, cellulose, glycogen, pectin, and dextrin. Starch is a storage form of glucose in plants, whereas glycogen is storage form of carbohydrates, particularly it is stored in muscle and liver. And coming to heteropolysaccharides, if all the monosaccharides, more than 10 monosaccharides, are of different category, we call it as heteropolysaccharides, and they have, the examples includes heparin, hyaluronic acid, chondroitin sulfate, keratin sulfate, dermatin sulfate. Now, let us see about the classification of lipids. Lipids are the compounds, organic compounds, which are insoluble in water, but it can be soluble in the aqueous solutions like benzene, ether, and chloroform. Coming to the classification of lipids, they are classified into 3 categories: simple lipids, compound lipids, and derived lipids. Lipids are also known as the esters of fatty acids with alcohols. Coming to simple lipids, they will have only fatty acid plus alcohol in its structure, right? And they are again classified into 2 types, fats or oils and wax. Next category is compound or complex lipids, where they will have fatty acid, alcohol, plus it contains an additional group like phosphatidic acid, carbohydrate, and proteins. If the additional group is a phosphatidic acid, we call it as phospholipid, and if the additional group is a carbohydrate, we call it as a glycolipid, and if the additional group is a protein, we call it as lipoproteins. Now coming to phospholipid. So they exactly contain the fatty acid, alcohol, and phosphatidic acid as is an additional group. Based on the alcohol, these phospholipids are again classified into 2 types, glycerophospholipid and sphingophospholipid. Coming to glycolipid, where additional group is carbohydrate, so their alcohol is sphingosine. We also call it as glycosphingolipids. And the examples for that are cerebrocytes, gangliosides, globocytes, and sulfatides. Now coming to lipoproteins, where your additional group is protein. Here they are again classified into 4 types, chylomicrons, HDL, LDL, and VLDL. Now moving on to the classification of proteins and amino acids. Proteins are the polymers of L-alpha amino acids, where they have 2 functional groups, amino group and carboxylic group, attached to the single carbon atom. Based on the structure and chemical nature, and based on the polarity, based on the metabolic fate, and based on nutritional classification, amino acids have been classified. So based on the structure and chemical nature, they are again classified into 7 categories. Aliphatic side chain, wherein it will have glycine, alanine, valine, leucine, and isoleucine. Coming to hydroxyl group containing amino acids, we have serine and threonine. And next is sulfur containing amino acids, wherein we have cysteine and methionine. Coming to acidic group amino acids, we have aspartic acid, glutamic acid, its amides, asparagine, and glutamine. Coming to basic group amino acids, they are lysine, arginine, histidine. Coming to aromatic amino acids, they are phenylalanine, tyrosine, and tryptophan. And coming to immunogroups, they are proline. So these are the 20 amino acids which can able to synthesize the structure or involved in the formation of protein. So this is the first category of classification. Second is based on their polarity. Again, they are classified into 4 types. One is non-polar, and second one is polar with no charge, and polar with positive charge, and polar with negative charge. And the third type of classification is based on the metabolic fate. The product, that means after the metabolism, the products, whatever they are released, if they are related to the carbohydrate, that is glucogenic, we call it as glucogenic. And if the products are related to the lipid metabolism, we call it as ketogenic. And sometimes both the products will be there as an end product. We call it as glucogenic and ketogenic. Now coming to the fourth type of classification, that is nutritional classification, wherein they are again classified into 2 types, that is essential and non-essential amino acids. Essential amino acids are those which cannot be synthesized in our body and has to be supplied through diet. So overall, out of 20 amino acids, 10 amino acids are essential and 10 amino acids are non-essential. Among these 10 essential amino acids, like arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan. These are the 10 amino acids which are essentially important. Among this, arginine and histidine are semi-essential amino acids because they are not synthesized by the growing children. So, this is the third category, semi-essential. Proteins are again classified into 3 types, simple, conjugated, and derived. And these simple are again classified into 2 types based on their structure, that is globular proteins and scleroproteins. Under globular proteins, we have albumins, globulins, glutens, protamines, prolamines, histones, and lectins. Under scleroproteins, we have collagens, elastin, and keratin. Coming to conjugated proteins, where they are an additional group. Like proteins, when they are combined with nucleic acids, we call it as nucleoproteins. When they are in contact with carbohydrates, if the carbohydrate content is less than 4%, we call it as glycoproteins. And if the carbohydrate content is more than 4%, we call it as mucoproteins. When proteins are combined with lipids, we call it as lipoproteins, and phosphoproteins, chromoproteins, metalloproteins. Coming to derived, these are the simpler or hydrolyzed products of simple and conjugated. And they are primary and secondary. Coming to primary, we have coagulated proteins, proteins, and metaproteins. And the secondary, we will have proteases, peptones, polypeptides, and peptides. So peptide bond is a linkage where proteins, it will interlink all the amino acids which are helpful for the synthesis or the formation of proteins. So coming to nucleic acids, wherein we are going to learn about the structure of DNA and RNA, which is the molecular basis of life. Thank you.